I think that we have fundamentally misunderstood, or at least underestimated, the way that science and communication are connected to each other. I think they're connected in very profound ways, and I think that failing to understand that connection has caused all kinds of problems in both communicating science, teaching science, and even doing science, doing research. And so what I'd like to do in the first part today is I would like to share with you some experiences that led to my building what I call a new model of science communication. In teaching and in practical experiences has been really effective. And so I've done this many times at lots of different institutes. I'll warn you in a moment about a little problem that we'll have. Okay, so like I said, I think I have a manifesto, and that is, I think we've misunderstood this connection, that if we do understand it, we can improve both the quality of the communication and the quality of research. And this is important because my job today is to convince you that it's worthwhile investing some time in communication and caring about this. And I know that from having worked with scientists for 25 years now, that the only way to do that is to convince them that it will help their research. So that's my goal. And I really hope that I can fulfill that promise today, and you'll be the judge of whether I actually do that. The structure of today's talk will be the following. The first part, this theoretical part, is I'm going to tell you a story about how I had to deal with all kinds of problems in science communication, which eventually forced me to make this theoretical model and to try it out and to use it on all kinds of ways. After the break, in the second part, we're going to do a very short thing where I'm going to show you how we can use tools of communication to play games, sort of, and how that can directly influence the way that you think about science and research. And the third part will be very practical, because this model that I've developed, this theoretical model, has some very specific implications for the way that we teach science and learn science, and then the way we communicate it. So that's my plan. And like I said, the first part is going to be kind of a personal story, because I found that that's the easiest way. What I need to try to get you to do is to see a structure in your head. And I'm going to try to help you build that structure through this story. So when I started to write about science, I discovered something very surprising, and that is researchers have problems, even though every day they have to communicate in all kinds of ways. They give talks. Every part of your career is based on a communicative function, like writing papers, giving talks, posters, writing a thesis. And I was very puzzled that people had so much trouble, because where I come from in the United States and in the Anglo-Saxon culture, communication is a part of our curriculum, and so we get trained. And in Europe, generally, it's not part of the curriculum, and people don't teach it very systematically. Another problem is that people obviously have to speak in English a lot of times, and there are some second language issues. But what surprised me the most was not those things, but it was that scientists seem to have a lot of difficulty when they have to describe their own work. And I tried to sort of analyze those problems, and they fall into certain categories that we recognize. So for example, a lot of times people fail to distinguish the main idea from details in a text, or they don't give you any context for a piece of information. There are all kinds of gaps or jumps in logic when people are talking about their work. And the biggest problem is, for some reason, our brains aren't built really to do language very well. And it's really hard to find the clearest way to explain what's in your head in language. And the result of this is that sometimes, just because a piece of communication is unclear, people misinterpret this and they think, oh, scientists are hiding something, or they're doing dangerous things. They don't want us to understand. They're being complicated on purpose. And usually that's not, obviously not the case at all. Usually it's just a failure of communication of some kind. I had the chance during my job to talk to a lot of Nobel Prize winners, about 15. I've been able to sit down with them and have rather long, intimate discussions with them. And I discovered that those people and a lot of other really great scientists that I know actually are very good at communicating and explaining their work. And they can explain it to practically anybody. And for me, this raised an interesting question, and that is there a connection between great science and great communication? What is it about being a great scientist that makes you a good communicator? And the opposite question would be, if I can help someone with their communication, is something also happening with their science? And so to solve this question, I really spent a long time. I tried to take a scientific approach to investigating what the connection is between these two things and how it works. And my method was kind of a genetic method. So in genetic science, we study organisms that have defects in genes, and those defects tell us something about the healthy function of those genes. And so I thought maybe we could do the same thing for communication problems. Maybe by studying problems that people have when they're communicating, we can also learn something about how you're supposed to communicate and also how we're supposed to think about these things. And this analysis, which took 4 or 5 years of really intense work, it led me to some surprising findings. And as I talk to you about these things, and as I tell you this story, it's really crucial that so most of my examples are going to be from the field that I am most familiar with, and that is the whole spectrum of kind of molecular biology, biophysics, and structural biology, and chemistry, genetics, evolution, and so on. But there were parallels to all of these things that you will find in your own work. And you need to try, if possible, to actively listen and actively try to translate these concepts and these examples into things that you're familiar with. Because that's the only way that any of this will sort of transfer into your field. In a way, it's good that I'm going to talk about biology, because most of you are not biologists, and so you'll struggle maybe with some of the same kinds of things that I struggled with. Now, one thing that came out of this analysis that surprised me a lot was I discovered that between experts, experts have communication problems with each other, particularly if they're talking across disciplinary boundaries. If you're talking to a chemist, or if you're a physicist and you need to talk to a biologist or a chemist, you have certain kinds of problems, and actually those are very similar to the problems that people have when they talk to non-experts. And so again, I discovered that there's a logic to those problems. There's a reason for it. There's a kind of mechanism that underlies it. And if you can understand how that works, you will improve not only both scientific communication, the way that people talk about their work, but you will also actually make them better scientists. And it's happened so many times that I'm truly convinced of this. And again, I don't know anything about physics, but I think we'll still be able to find some parallels. And I've talked to enough physicists and helped them with their communication problems enough to believe that these things are transferable. Okay, so to get you into this story, I'm going to give you an example of one of the very first stories that I had to write about in science. And this was a theory that scientists had about the structure of cell membranes. Now, here's a picture of a cell, an animal cell, and here's a picture of the membrane. And if you zoom in on a little section of this membrane, you'll see that it's made of fat molecules and proteins and so on. And oftentimes to fulfill a function, like to receive a signal from the outside, a cell needs to group a bunch of different molecules together into a sort of collection. And the question was, how does it do this? And scientists at our institute had a hypothesis that inside the cell, as it's building molecules, it creates what we call rafts. Rafts are those things on rivers, you know, Mark Twain and Huckleberry Finn floating down the river on a collection of wood, and so the cell makes these things and it transports them as a unit, kind of a prefabricated unit, out to the cell surface. And this was an idea, and various experiments had been used to kind of demonstrate this, but it hadn't been proven before. And so some scientists at my institute, using tools from physics, actually figured out a way to test this hypothesis. And the question was, how big are these things? Or do they really exist? Do these collections of molecules and lipids as structures, do they really exist? Or are all of these pieces independent and kind of working on their own? And what they did was they used a method called optical tweezers, which is a laser focused on, they took beads, microscopic plastic beads, and they coated them with antibodies. And those antibodies would bind to molecules on the cell surface. So they would bind to these oops, didn't want to do that. They would bind to these molecules. And when that happened, the tweezers they would track the movement of the beads and measure very, very precisely whether it moved easily or whether it took a lot of force to move the bead. And they could measure, and the scientist who told me this story, he compared it to fishing. He said, I come from Kansas where we fish. I spent most of my childhood in a boat fishing. And he said, imagine that you throw your line into the water and you catch a really big fish. Well, that fish is hard to pull in. And if it's a small fish or there's no fish, then you can pull in your line very easily. And I thought that was nice, a really nice way to explain it to me. He used a metaphor, very kind. But there was still something I didn't understand about this story. And the problem that I was having was that the last time, I hadn't studied biology. And so the last time I studied cell membranes, just a moment, I'm having trouble with my Zoom view. Page display, cell page view. Okay, we'll try this, go back into read mode and it should work. Sorry about that. Let's try this. Nope, that's not right either. So, okay, here we go. Okay, so the last time I'd studied cells, I had seen membranes as these big fat lines around cells. And I thought, okay, a cell it's like a wall around a cell. And how could you possibly pull a fish through a wall? I just didn't get it. And the scientists and I talked about this for a long, long time. And finally, the scientists realized that I had this wrong idea of I didn't know what a membrane was like. He said, don't think of a membrane like a wall. Think of it like a soap bubble. You know, children blow those bubbles on the playground, and think of it like that. And because it's made out of fat molecules, like soapy molecules. So when I thought of it like soap, then I could realize, okay, maybe you have something floating in the soap and then you can pull it around like a fish. I could understand that. And the problem that we had was that the scientist had a model of a membrane in his head, which is called the fluid mosaic model. And it shows that it's made of these fat molecules, it's soapy, and there's things floating around in it, and they can either be stuck together somehow or they could be loose. And so that was the issue. And what was really interesting to me was our communication problem lay in the fact that the scientist had this model in his head, and I didn't have that model. And so the other thing that was really interesting was the scientist never thought of explaining to me this model, because every scientist knows what a membrane's like. And it turns out that the things that people know the best are oftentimes the things that you need to know the most to understand a story. And they're also exactly the things that nobody tells you. because everybody's supposed to know those things. Well, I didn't. And so this was the first clue or the first hint that I had of what was going on here. And what I discovered was that scientists have what I call the inner laboratory. So of course you have a real laboratory where you go and you pour things back and forth and you use electricity and you use all kinds of instruments. But you also have a laboratory in your mind, which has things like what a membrane is like in it. And to understand a scientist, I have to see what's in that laboratory. We have to have kind of the same stuff there. And if there's something missing, I can't understand. Obviously, it could be something really simple like what a word means, but it's usually something a little bit more complex, like what is a membrane like, or what model of a membrane do I have? And when I began to understand this, I realized that there's a lot more than just facts and definitions in this laboratory. And I'm going to show you a little text now, our first kind of real example of science communication from biology. I'm sorry, but we're going to work on this together. And I'll show you how it works, because this text shows us a lot of different things that are in that inner laboratory. So this is a real key concept, the concept of inner laboratory. I'm going to be talking about it all day, so remember that. And you need to explore your own, and we'll talk about that later. I'll show you how to do it. But okay, so here's a text. This is a text about when cells get signals from the outside and what happens then. So the text goes, cells constantly produce and degrade the molecule beta-catenin. Normally, it is bound to a complex that is targeted for destruction. Signaling by Wnt blocks the formation of this complex, leaving higher quantities of beta-catenin. That means it can enter the nucleus and activate target genes. Now, the first time I heard this kind of thing, I thought, okay, I understand most of the words in that, but there's a lot going on here. And what's going on is you have to know several different kinds of things to understand this text. The first thing is there's several different kind of models or patterns that you need to see. So you have to know about the terms, what the terms mean. You have to know things like what is beta-catenin, what does produce mean, what does activate target genes mean. You also have to see how these different terms are related to each other. And part of that has to do with the geography of the cell. And actually, this is a sequence of events. So when the scientist says this, it's like he's watching a little film in his head. And what I need to do is I need to see that film. So let's take it apart and see what's it made of. Well, the first thing is he has a picture of the cell in his head, which has, here's the inside of the cell, here's the nucleus, here's this complex he's talking about, here's this thing called beta-catenin, and here's a signal from the outside. So he sees all of this as a sequence of events. He sees a signal coming, it activates something, it stops the formation of this, whatever this destruction complex is, it releases this goes into the nucleus, and this does something. So there's a map and there's a sequence of events. All of these things are part of a much more complex sequence of relationships between molecules and how they activate each other and how they influence each other, which we can draw also this way. This is a much more complex view of a signaling sequence like that. This is just shows the part of that system that we need to understand that story. And this is a much fuller picture that gives you a view of more things. Now, both of those are just different ways of representing a real situation in the cell. They're models, okay? And signaling in cells, this biochemical interactions that make everything happen is actually a lot more complicated. So here's another version of that signaling molecule, which is another way of drawing it. And you can use it in different ways. What's missing here is the geography of the cell. We've lost that. But and what's also missing is this kind of sort of very straightforward story, like this does this to this and that does that to that. We see that everything is much more interconnected to each other. And here's another model of the same thing, which is an analysis. It's an algorithm. It's a mathematical model which is used to predict where you can perturb that system and have specific types of effects. So I've shown you 4 different models of basically the same thing, and each one of them does something different. And scientists can read, maybe different scientists use different ones and read them in different ways, but you know that all of them are oversimplified in a certain way. And what scientists learn is not only what these models look like, but they also learn how to unpack them. And so if we look at this, this process of unpacking communication involves a lot of things, a lot of these invisible things that we don't really talk about, agreements and assumptions. So for example, it just starts, cells constantly produce and degrade the molecule. What type of cells are we talking about? What species? Produce, what does that mean? That's a very, very complex series of events. There's probably 50 steps involved. Molecule, DNA, RNA, protein. Beta-catenin comes in different flavors, in different sizes, in different forms, and each person's version is a little bit different. When we're talking about complexes, do we know all the components? Do we know how they're built, arranged? Do anything of these things matter? Do we need to know those? Well, scientists can tell you which ones are important and which ones aren't important. I can't, because I don't understand the system. And the other thing that's important to realize is that all of these are based on these models, but what if the models are wrong? We constantly, I mean, science is a process. It's not a process of discovering truth. It's a process of modeling something and then interrogating that model with data and then fixing the model and making a better model, constantly making better models, okay? And we know that they're wrong, but they're still useful. Anyway, it doesn't matter. So, and the other thing is that this story, this little story about a signal in a certain kind of cell is actually part of a whole bunch of different kinds of models. So there's evolution, there's cell biology, gene expression, biochemistry, structural, but all of these fields, somehow this text is related to all of these fields. And I mean, you could go crazy, right? Because you think, my God, to understand this, do I have to know everything? Do I have to know all of those things to understand this text? Well, if I know something about all of them, I will understand it. But the real question is, what do I really need to know to understand it? Now, I'm going to switch and I'm going to give you another example of the same kind of problem. I'm going to show you something. And the thing that I'm going to show you, I guarantee you that each of us will see something different. We'll all see the same thing, but on the other hand, we'll all see something different. So here we go. Now we have background noise, sorry. This is a chessboard, and I guarantee you that a person who plays chess, this looks completely different than someone who doesn't play chess. First of all, and a person who plays chess really well will see something different than another person who just plays kind of like me, low average, I would say. And the reason why this board looks different to us is because of what's here in this inner laboratory of chess. Just like we have an inner laboratory of chess, we have an inner laboratory of science, right? Okay, but in chess, we can actually say what the things are that should be in that mental model or that mental laboratory. So we need to know what the pieces are. We need to know the rules that govern the movement of each piece. We need to also know some basic things, like you have to know that one person plays, white plays, and then black plays, and then white plays and black plays. You need to some sort of, you also kind of need to know the goal of the game, which is interesting because the goal is to capture the other king, and that never happens in the game. The game always stops right before that happens. So it's an interesting situation. And if you know those things, then you can answer some important questions. So for example, is it possible that this is a real chess game, or is there one piece too many on the board? It could be. Or what was the previous state of this system and what will happen next? And all of those things are also true of scientific models. So if we have a scientific model, the goal is to look at a system in a certain state, to know what the components are, to know how they interact with each other, and then to be able to project that system backwards and forwards in time and to say what it used to be like and what it will be like next. Okay, and so this is in your mental laboratory, but all of that stuff, if you're just looking at the board, if we're looking at the board together, it's invisible. So I was having all kinds of trouble understanding science and writing about science because of these invisible things, and I needed a name to talk about invisible things, so I picked one. I call them ghosts, and I think it's a good name because a ghost is something invisible, but it's highly disruptive. And in this case, we have ghosts that are invisible. That is to say, they're not on the board. The rules are not here. You can't see them. Or in a text, the rules are not in the text. All that knowledge is not in the text, it's in the background. So ghosts, and there's lots of different kinds, and we're going to talk a lot about this because I keep finding new kinds, and you'll probably find new ones yourself. So they can be pieces of information, or they can be concepts, or they can be patterns of connections between ideas, or they can be all kinds of other kind of knowledge, and they're not in the text or the talk or the image or whatever. But to understand what it means, you have to know those things. So for example, all those diagrams of a cell I showed you, I mean, if you didn't know that this was a cell and this was at something microscopic, I don't know what you would think I was showing you. Or all those little balls, maybe those are like people's Facebook connections or something. Again, you have to know certain things to understand what these means. And what I found was really interesting and crucial is that you must learn to see that these things are there, and you have to also be able to identify them. Otherwise, it's really hard to learn science, and also it's really hard to communicate it. Now, I think people instinctively know this to a certain degree, but there's a lot of this that isn't instinctive. And I'll use another example to show you how bizarre these ghosts can be. So, and one last point about this is, sorry, I changed my slides. You should never do that. So another thing is that these ghosts, they not only disrupt communication, but they disrupt research, because ghosts are hidden knowledge or assumptions that you may not be aware of. So for example, when a scientist is talking to me about the membrane, he may not consciously realize that he has this model that dictates what he says and how he describes it to me. He may not be aware of that, but it's powerfully shaping how he sees a system and what he sees in the data from his experiments. And so they can influence research. And the next thing that you need to know is, if you want to find them and expose them and make them visible, the best way is through communication. And I'll give you another example of that, which to me is one of the greatest examples that you can possibly find. And this has to do with, so we've been talking about architecture, patterns and sort of models. And so let's translate this into a real building, a picture of a building. So if we're going to talk about architecture, the architecture or the structure of your inner laboratory, let's look at another kind of structure, and that is look at the structure of a building. And this is a building here in Berlin that our architects built. And this diagram, even they can't read this because this has everything in it. The blue things here are the walls of the building. And you can see also the electrical circuits, and you can also see the ventilation and the water and just and the space, everything is in here. And they had to make this specially for me by combining all these things. And when they actually build something, they peel it all apart and say, okay, we're going to first put in the walls and so on and so on. Okay, now that's way too complicated. So let's look at a simpler kind of architecture. And my sister lives in Kansas, and she has a nice house. And so I had her draw a floor plan of her house. I just told her, Karen, I'm sorry, her name's Karen. I know that name is loaded now, but she's a very nice person. Karen, draw me a picture of your house. And so she drew this diagram, which could be helpful because sometimes I stay there and in the night you may have to get up and walk through the dark, and it's good to know where things are. So you can see the floor plan of the house. So there's a garage, there's a kitchen, a living room, There's a master bedroom, and then there's the boys' room. My sister, who's much older than I am, has grandchildren, and she has, in fact, identical twin grandchildren, which will be important in just a moment. So she drew this plan, and it was very nice. I told her it doesn't have to be very accurate to scale, and this is part of an experiment. So then I told her, I want you now to have your grandchildren, the twins, draw the same house. And you have to know, first of all, the twins are 8 years old. We're 8 years old at the time. And secondly, they're identical twins because I needed a control group. Okay, so what do you think a house looks like to an 8-year-old? What do you think an 8-year-old's view of space and rooms and houses is like? Is it like ours? Is it like an adult's version? Or is it different somehow? You're going to see now. So this is the first twin. He drew this house, and here's the kitchen. So if you compare it to my sister's drawing, it's kind of the same. So there's a living room here, the kitchen here, there's a bedroom here, closet, something, bathroom. Everything is relatively kind of in the same space to each other. But I call this the octopus house. It has all these kind of tentacle-like things, the halls and the corridors that connect things. And now we'll look at the control, which is his twin brother. This is more the scientist. Again, so if you compare that to my sister's drawing, again, you see that things are kind of in the same place relative to each other. And the sizes are also kind of okay. And you have all the, you can see a little person in here. I've never seen that before. Every time I look at this, I see something new. He also has the backyard, and he says, P.S., it's massive, which is true. They have this huge yard. Anyway, but if you look at these two pictures, you discover that while they do have some similarities to reality, to my sister's version, they're also quite different. And what you learn is that there's two things about houses that children don't really understand. There's two rules about houses that they don't know. And one is they don't know that the wall of this room is the back of the wall of this room, and that this wall is the back of this wall. And you can do this experiment. Take a small child and say, go into the next room. I'm going to knock on the wall, and guess where you'll hear the sound. And they'll be very surprised by this because they really don't understand space this way. And the second thing that they don't understand is that it all has to fit into the shape. If you walk around the outside of the house, it has this shape, and it all has to fit into that shape. And they don't know this. And so we see that children have a and this explains some things. Like when I was a child, I always used to wonder whether maybe there were some secret rooms or secret passageways in my house. And I didn't know that you could actually measure the rooms and figure that out. I thought, because there's all this mysterious space, there could be anything in here. And maybe you had those kinds of ideas also as a child. Well, the reason is because you don't see space the same way as an adult. And we would never know that. I mean, the child can walk around the house and he can find everything he needs. But the only way that we know that is by having him draw it. So what this teaches us is that children organize their knowledge very differently than adults. They lack two rules that every adult knows. And the only way that you find this out is by making them draw or communicate. And the same thing is true of science. Your inner laboratory is different than your colleague's inner laboratory. It's organized differently. It has different stuff in it. Some people have very neat and highly organized inner laboratories. Others have very messy inner laboratories where you can't find things that you need. And the only way to see that structure is to communicate, just like the only way to see the real difference between a child's understanding of space and an adult's understanding of space is to have them communicate that somehow through a drawing or just a text. You could hide it. It would probably be less apparent. So in fact, this tells us something even more important, and that is it tells us that the real function of science communication is science communication is a chessboard for science. So it's the board that we play science on. And a chessboard is there so you can learn how to play the game. It's there so that you can study the games of the masters. It's also there for practice, so you can try your hand against ever better players. Because again, what we're arguing about in science and what we're trying to solve is we're trying to fit all of this data and all these measurements we have to models. Those models, there's no place in the world where you can go, there's no book or no website where you can go and say, I want to find the perfect scientific model for everything. They're dynamic. We're continually changing kind of the rules, and we're finding new pieces for the board. And so what we're doing is negotiating our models. And if you don't communicate, you can't do that. And you also can't see whether a person is a good player or a bad player, or how they have this inner laboratory organized at all. So this is really crucial. Scientists have, to kind of summarize, scientists have this rich inner laboratory where really science happens. It happens also in the lab, but you can go into the same lab. You could go into a laboratory that existed 100 years ago, and you could use some of the same equipment, but what you would do with that would be completely different because the inner laboratory is different. And 50 years from now, we may still have laboratories which have much of the same equipment, but what we will do and how we will understand things and the results will be different because the inner laboratory is different. Without communication, it's impossible to see how somebody else's knowledge is organized. And I believe it's also impossible to see how your own knowledge is organized. And I'm going to show you an interesting couple of examples of that later. So, and if we're going to communicate science well, then the first thing we need to do is we need to understand the patterns and those structures that are there. We need to see what's in there, because, and the next thing is you have to guess, okay, if I'm going to talk to you about biology, I need to guess a little bit about what you know about biology and how if you have an inner laboratory, and if not, I need to help you start to build one and to organize it. Now, I've talked to you about these ghosts, and I've talked to you about them in the context of science. It's all of this invisible stuff that explains what things mean. And what things mean in science has to do with models. That's not only true in science, it's also true in other parts of life. So ghosts make a mess of all kinds of other communication. For example, I went to the United States during the election, which was a big mistake. And I talked to people and they would tell me things like, I voted for Donald Trump because he's going to make America great again. And I really tried to understand what they could possibly mean. And I tried to figure out how their knowledge was structured, what would make them say this. And it was just hopeless. But in science, what I found is there's certain rules about how we do things in science that means it's possible to identify the ghosts. If you show me a text, I can show you where there's going to be ghosts. Even if I know nothing about your project, I can show you where they're likely to be ghosts, and I can also identify why I can't understand something. So Martin, who's one of the participants today, he gave me a text, and I looked at that text, and there's a lot I didn't understand, but I could always tell him why I didn't understand it and what the reason was. And that's really important. So I'm going to show you now another real life example. And we're going to use this text many times because I'm going to use this to actually show sort of several things. Now, this text was written as a press release by my biology institute. And I really hope that it's really hard for most of you to understand. If you understand it well, then no, anyway, good, congratulations. But anyway, so this story is called Rewrite the Textbooks. Transcription is bidirectional. And we're just going to, I'm not going to explain anything. We're just going to look at this text first. And genes that contain instructions for making proteins make up less than 2% of the human genome. Yet for unknown reasons, most of our DNA is transcribed into RNA. The same is true for many other organisms that are easier to study than humans. Researchers have now unraveled how yeast generates its transcripts and have come a step closer to understanding their function. The study redefines the concept of promoters, the start sites of transcription, contradicting the established notion that they support transcription in one direction only. The results are also representative of transcription in humans. Now, you should know this was intended as a press release. That means my institute, after I left, this was not my work. They sent this out to newspapers, hoping that the newspapers would print this. And presumably, if you do that, you hope also that people who read it will understand it. And that was not the case, usually. And again, this is when it would be nice to have a live classroom, because I could ask for a show of hands how many people understood something in this text. We could even talk about what you understood. I can make some predictions about what you understood, but we'll see about that. But this brings us to a really more basic question, and that is, what's the goal? Why would I show you this text? What are we trying to communicate? And is there a test that we can perform to measure whether that communication has been successful or not? Again, this is something that it would be nice to have time to think about and discuss as a group. But I'm going to just kind of set the table for you and jump ahead, and we'll see some more examples of this later, and we can actually discuss it. But generally, when I show people a text like this, I'm not trying to get them to memorize it, okay? I don't want them to memorize a list of sentences or a list of facts. Instead, I'd like them to grasp a small set of ideas. I want them to understand something. And one way to test whether they've understood it is to say, okay, tell me this story in different words. And if they can find 2 or 3 different ways to tell the same story, and they're all right, in my opinion, then I've probably been successful. And the other thing that I want them to be able to do is I want them to be able to use those ideas somehow. I want them to understand the implications of those ideas, or to see how they might be important or relevant. And when you do this, it turns out it's more important to reproduce the pattern than to really get people to reproduce any specific fact or detail of this. Now, I'm going to give you another example of this, and I'm going to show you something that you all have learned. Now, I'm talking to physicists, and so you probably, with biologists, this is a great test, because biologists, none of them know this. And that is, when you were in grade school, like me, you probably learned about the solar system, and this is the way the French learned the solar system. It has Le Soleil at the middle, and Jupiter, and La Terre, and the other planets, and you can use this to learn French. But anyway, so you learned, and we learned, in fact, we learned the names of the planets, and I can still tell you the names of all the planets. Mercury, Venus, Earth, Mars, Jupiter, Saturn, Uranus, Neptune, Pluto. Order the planets going outward from the sun. And I can tell you those because we learned a little sentence. My very educated mother just served us 9 pickles: Mercury, Venus, Earth, Mars, Jupiter, Saturn, Uranus, Neptune, and Pluto. And so that's fine. I learned the names of the planets, but I had a really clever teacher who also wanted us to be able to use this information. So for example, my parents bought me a telescope, and he said, the coolest thing to look at in the sky at night is the planets, because they're close. So can you go outside at night and find all the planets in the sky fast in 3 seconds? Some of you who are physicists who have hobbies as astrophysicists or whatever, or like the planets, can probably do this, but most people can't. And that's because they learned the system, and maybe they learned some facts about it, but they didn't learn how all the pieces were connected into meaningful patterns and how to use that information. So if you learn this the right way, then you can actually answer some interesting questions. So for example, the first challenge is, can you go outside at night and find all the visible planets? You should be able to do it really fast if you really understand how this system works. And if maybe there's 1 or 2 of you who don't know the trick, but I'll show you the trick. I'll show you the trick using my desktop here. I don't know if you can see this, it's kind of small. So I'm going to rebuild the solar system on my desk. Here's the coffee is the sun, obviously. So, you know, it's at the middle. And then we have some planets moving around the sun like this. And the first thing that you need to know about the solar system is that all the planets move around the sun on a plane like the table, okay? So you see here, they're all in a line with each other. And it's, so there's not one that goes around the sun this way, around, over and under the table. They're all sitting flat on the table, okay? And the next thing you need to know about this system is that the Earth rotates. I'm here. I'm going to show you where I am. Here I am on the Earth, right there. I'm on that spot. And the Earth axis is perpendicular to this plane, so it rotates like this. So if you know those two things, it tells you that. Sorry, somebody's. Yo estoy atendiendo un seminario. Needs to turn off their microphone. Sorry. Okay. Anyway, so if you know those two things, then you know that all you need to do to find the planets is go out into the sky and look for that one plane that represents the table surface. They're all the planets will be on that one line. And the way to find it is really easy because, again, the Earth's axis is perpendicular to this. That means that the sun is also on that line. And so all you need to do is to know where the sun goes up and down, where it rises and sets, and the line that it goes. And most people know this for their neighborhood. They know the sun's going to be there. It's not going to be over there somewhere. So find that line, study that line, and you look at it and you'll find all the visible planets. It's really easy. But in order to use this model, you have to see how it's connected to each other. And that means you have to put your mind into different positions in it. You see the connections and you put yourself on the Earth here and say, okay, what does the system look like from the Earth? And if you try to figure out things like, for example, why didn't people know 300 years ago or 400 years ago that the Earth travels around the sun rather than the other way around? That's because you have to understand what this system looks like from the Earth without that model, and how people constructed that model based on their observations. Now, here's a case where for sure there's applications to physics, because whenever I think of physics, I think of the atomic model, and I think of, for example, I see it in a certain way. There's the protons and neutrons at the middle, and then there's these electrons in clouds or shells or whatever. What does that system look like from the point of view of an electron? And you know, I know this, you know, uncertainty principle, and that's about the limits of my physical knowledge. But put yourself into that system, or put yourself into a cell and look at the cell from the point of view of one molecule, or from the point of view of different parts of that system. And then you can really see how it's put together, and you can start to use it. And you can also generate questions using this system. For example, you can find all the visible planets. You could also say, we know that the sun rises and sets 24 hours. It's a 24-hour cycle. The moon also rises and sets. How long does that take? Well, I'm not going to give you the answer. I'm going to challenge you to, if you don't know, to find that out on your own. But again, there you need to know one additional thing, and that is you need to know that as the sun turns, the moon is going around the Earth at a different pace. So if it were holding still like the sun, then it would take also 24 hours. But the moon does not hold still. It's also moving. And if you know those two rates, you can calculate that. And then put yourself onto the moon and ask yourself, how long does it take for the Earth to rise and set? Oh, the Earth doesn't rise and set on the moon. You also have to know something there. So again, if you understand the system, you can put yourself into different places, ask yourself different questions. And again, the question is, what are we trying to communicate? It's helpful to maybe know the name of the planets, you know, in case I want to go to Mars. I know that it's closer than Jupiter, but it's more important to understand how the system works. Now, if we go back to this text, the question that I'm going to ask you is, what is in this text? Have I given you the information that you need? What ghosts are there? And so on. And so if you just look at this very simply and say, okay, if a person's a biologist, then they have a sophisticated inner laboratory and they already know how all these pieces are connected to each other. So I can tell them the story one way. If you're not a biologist, I have to tell it a slightly different way. And I'm just going to ask a really simple question like, genes that contain instructions for making proteins make up less than 2% of the human genome. What is the connection between that fact and the next fact? Yet for unknown reasons, most of our DNA is transcribed into RNA. And if you look at this, you see that there's something missing. What's missing is what we call the central dogma, or a model of the connection between DNA, RNA, and proteins. And the model says DNA makes RNA, makes proteins. So if you know a couple of things, like genes are made of DNA, and then you can put those two sentences together. If you don't know that, then it's a ghost and you won't understand the story. And then we have the word transcribed, and then we have yeast generates its transcripts. What's a transcript? Well, maybe something that's transcribed is a transcript. So maybe that means RNAs, but that's something I have to do in my head. It's not here in the text. And then later we say the start sites of transcription. What is transcription? Again, so you can actually imagine that I'm learning all of this new from this text. How would I see the world based on this text? And you can actually make a map of this. Okay, so we learn that genes encode proteins, and we learn that they're a small part of the genome, and we learn that most are transcribed into RNA. That's one set of ideas in the first sentence. Then we learn that yeast generates transcripts that have functions. Another interesting, wonderful fact. Then we learn that promoters are start sites of transcription that was previously believed to happen in one direction, and now it's been contradicted. Another set of facts. If you're clever and you're willing to do some work, then you could look at these three and you could see that all of them contain the word transcript. And maybe you could then combine those pieces of information around this concept of a transcript. But again, there's lots of things missing. Now, a scientist, a biologist will be able to understand instantly that text because the biologist has a different map in their text. And all of these concepts have to do with a topic called gene expression. And I made a map of what I know about gene expression. I'll show you this later. It's a little tool that you can use to draw these things. And this is my map of gene expression. And it's pretty complex. There's lots of parts. It shows how they're connected to each other. And I highlighted the things in that text and showed you where they are in the map in my head. So these pieces, this is where they are in my map, okay? And because I have that map, then I can understand the text. If I don't have that map, then I have a problem. Then I'm trying to make one as I read. And whenever you communicate to someone, you're either giving them information that they're putting into an existing map, or you're helping them build a map. And knowing that that's the case can completely change how you go about the task. So if I'm trying to communicate patterns of meaning, then what I need to do is I need to see how ideas are organized in my head in patterns. I need to make a guess about what you know, and then I need to find the simplest way to help you reconstruct this map that I have in my head. Now, I'm not going to help you reconstruct that map. That's way too complicated. So let's analyze the story and let's see what's really in there. And when you do, you find out that actually to do this, you only need a few concepts to understand the story. And so I'm going to make a diagram of those concepts. You need to know that there's a thing called transcription machinery in cells, and it does several things. It binds to DNA. If you want, you can tell people it binds to these things called promoter sequences. I don't care. When it does that, it transcribes the DNA into RNA. Okay? And we used to think that it did that in one direction, and now we know it does it in two directions. And that's the whole story. You can talk, if you want, about what happens to that RNA. Some of it gets turned into proteins and stuff. But again, it's not essential to understanding the story. So this is the core of that story. And what's really interesting about this is that if you look at it closely, it's based on a pattern that everyone already knows. And you can find that pattern by looking at words like machinery and transcription. These are metaphors. So yes, it's a chemical process, and what we mean by transcription is a complicated set of chemical events, but it's actually a metaphor. And so let's look at the structure of that metaphor. And so we have a machine that does several things. It reads DNA, and that DNA is like a text, and it binds to it, and it understands it, and it transcribes it. And in our normal understanding of texts are linear, okay? So that means you can read them in one direction and they make sense. If you try to read them in the opposite direction, they don't make sense. And suddenly we found that we can do it in two directions. So that's the new. And if you see this pattern, it gives you a completely different way to tell the story. And this is going to be the basis of the rest of what we do for the day. So if I rewrote this text, and the first thing I did was I changed the title to "Our DNA Can Be Read Backwards and Forwards." And the way I started the text was, first of all, I didn't want to just give you a fact, but I wanted to put this into a context. So our cells specialize and cope with the challenges of life by producing different sets of molecules. They do this by drawing on different recipes in their DNA that they use to build RNA and then proteins. This process begins when a cellular machine scans DNA and locates the instructions it needs for a particular molecule. The machine attaches itself at the beginning of the code, reads it, and transcribes it letter by letter into an RNA molecule. Until now, scientists have thought that the recipes only made sense when read and transcribed in one direction, like this text. But scientists have now found that when it binds, the machine can often read and transcribe in both directions. I've told the whole story, and now I can even tell more. I can tell how they found this out, and I can also talk about what it means, its implications. So I've done several things here. I have engaged the reader by putting it into a context. I've said why cells would do this, or why the transcription machinery would do this, why it's important, why it's interesting, why it's necessary. So again, why is this relevant to why should we care that DNA can be read forwards and backwards? I've also used familiar patterns like recipes, machine, scanning, instructions, texts. I've also told the real story. I've highlighted the real story. I've put it in a place that has emphasis. So this tells what they really found, what they did. And if you go back and look at that other text, which you can do because I'll give you the file later, then you'll see that the information was also structured in a different way. So if I realize that I'm trying to get you to build this pattern, then there's 100 ways I can tell that story, because I want you to remember that pattern. And one way that I get you to remember it is by making it meaningful to you. Another way is by using familiar patterns. And we'll talk about that in a minute. And the other thing is really then to focus on the story. I'm going to show you some examples later of press releases from the FZU in which the story, they're trying to tell stories, but I'm not sure that they actually tell the real story or why and they certainly don't do any of these other things. They don't engage by telling its relevance or its importance. Now, if we do this, this is interesting because we've used a metaphor, and the metaphor grasps. So it's really important to understand that what you have in your inner laboratory is not reality. You have models of things and you have images of things, again, and some of these images are quite complicated. I really don't know what an atom is like. I have a kind of picture in my head, and you have a different picture, and you have some mathematical formulas to describe it and everything. Wonderful. But it's not reality, it's a model of reality. And that means it has patterns, and it uses metaphors, it uses images. When I see things, I'm magnifying it up to a scale of things that can be seen rather than the real scale, which is so small that you could never, vision doesn't even work at that scale. So if you try to draw anything, it's automatically wrong because you can't see it, it's too small. And we know that at the quantum level, weird things happen anyway. So anyway, what's in your head is not reality, but it's metaphors, patterns, concepts that you've built. And these are, and if you understand this, then you can play a game with it. And we're going to play some of these games in the second part, but I want to give you just a taste. So if our metaphor and our pattern is reading and writing, think of some different, so immediately this poses some interesting questions. So normally when people read my text, they say, okay, well, I wonder how the machine, it's a machine, machines don't think. So when this machine zooms in and grabs onto DNA, how does it know which direction to go? That's actually a really interesting scientific question, and you can ask it without knowing anything about cells or anything about chemistry, just based on the patterns in the story. Also, it implies, if we're thinking of a recipe book, well, if you can read a recipe book forwards and you can also read it backwards, that means there's at least twice as many recipes in the book. That's an important implication of this story that's really scientifically important. And again, to understand that implication, that means our cells have twice as much information in them, at least, lots more actually, but that's okay. And so as a non-scientist, you can actually draw a sensible scientific conclusion from this. Then you can start to play. Here's the game. The game is, if I understand the pattern of reading, what are some other things that we do when we read? Well, when I read, I skip some boring passages. I wonder if the transcription machinery, when it locks onto DNA, also skips boring passages. You could ask a scientist that question and they'll say, in fact, yes, it does. What happens if I misunderstand a sentence? Can I go back and reread it? Does that also happen in cells? Maybe, maybe not. Can I stop reading and answer a phone call? Can I mark a page so I can come back to it? Can I go back a few pages? Can I look things up? All of these are just playing with the pattern of reading. And so, but since I understand that pattern, now I can go back to my scientific system and say, could these things also happen in a cell? And in fact, many of them can. And you might not think of them, of asking these questions, if you're so immersed in your transcription machinery scientific model that you can't see that there's a more fundamental pattern and that you're using that pattern to construct a story. Well, then let's play with it. So that's the first game. And this game that you can play once you understand that your inner laboratory is metaphorical, conceptual. You can mix chemicals in your inner laboratory that you would never think of mixing in the real world. Or you can apply patterns that you don't apply. And again, we're going to do some more of this in the second part. It's in all of this, it's really important to connect the stories that we have to tell and the questions that we're asking in science to the models that lie behind them. And if you don't see that every single thing in science is, its meaning comes from models, then you waste an immense amount of time. So when I read a text that I don't understand, at least I know that everything in that text is there because scientists have this model of this system and they're trying to use it to understand how a material works or how things interact with each other or how to build something or how to understand a system. And that's governed by the models that we have right now. And those models are different than they used to be. So most scientific projects are related to a model. Either you're modeling a new system, you've got a new game that you've suddenly discovered, a new type of cell, a new organism, a new physical system, a new material. And you're trying to discover what the components are and the rules by which they interact with each other and how they form a larger system that evolves and develops. Or you have a model and you're challenging it by generating hypotheses. You've invented a new kind of microscope, or you've discovered a new level of resolution of something, and you discover there's new components and you're trying to see whether they fit into the model that you have. Or you're trying to extend a model to a new system, or you're trying to integrate a model that you have into a more general one. It took a long time for people to connect population studies to genetics to evolution, for example. It took decades. Or you're saying the model that we have of this system, we have data which suggests that it's not the right model, so we need a new one. If every time you taught someone, or if you read something, or if you learned something in science, if all the time you were aware of its connection to this network of models in your head and in other people's heads, it would save you immense amounts of time because you're being flooded with single facts like names of planets. And the crucial thing is to see how they fit into an integrated, interrelated system. And what we fail to do when we teach and we communicate is we fail to put things into these little, and they're not vast networks usually. Usually each story is a small network of things that we need to understand. And if you fail to do that, it just wastes everybody's time. So scientific projects are related to models. And the last thing I'm going to do is I'm going to show you how this relates to the bigger picture. I told you at the beginning, Nobel Prize winners are really good at communicating. And now I'm going to show you why. I'm also going to show you how to win your own Nobel Prize. I'm kidding. No, not really. Okay, so I'm going to take an example from biology. And the example is, I have a colleague who's working on the question, what is the structural basis, that means the chemical and physical basis by which a molecule called NF-kappa B is able to bind to a molecule of DNA in the cell. And that question is being asked in terms of different kinds of models. One kind of model is the way protein DNA chemistry works. Another kind of model is the fact that molecules build machines and the functions of these machines are determined by the components and how they're arranged. So we need to find what it binds to and how those work together in a physical and chemical way. The effect of these combinations and this molecule is to activate different genes in the cell. And again, this one, it's called a transcription factor here. And a transcription factor is a gene activating molecule. That means there's some really powerful molecules in cells, and they're able to go into the nucleus, and they're able to read the recipes there, and they're able to switch it on and create these RNA molecules. Now, when they do that, this helps cells to adapt and respond differently. And they need to change their responses as they develop or as environmental conditions change or during diseases, for example. And this process of development, which is guided by these transcription factors and all these other things, describes, for example, how many cell types arise from a single fertilized egg during embryogenesis as the embryo develops. And all of those things relate to how an organism's health depends on the very basic fundamental components that make it up. Now, this understanding how these things are connected, these are different levels of a problem. And the student is actually working on this at this level. He really wants to know the physical and chemical details of what's going on down there. But at the same time, he's actually also working on all of these other levels of problems. Ultimately, he is working on questions of how an organism's long-term overall health is connected to the little tiny stuff in our bodies. So this connects different levels of structure and thinking because, and it's important for him to know that this is connected because as he learns something about this, he's also learning something about all these other things potentially. And if he sees that connection, then it has many, many benefits. And this is how I'm going to close this part of the talk by showing you how these two things are related. These have scientific functions and they have communication functions. Most Nobel Prize winners won their prizes for work at this very tiny, tiny, specific, detailed level. But they found the right places in cells or organisms or physical systems or material science or whatever, that what they discovered would tell them as much as possible about these other levels. And the really smart guys see that as they're working on this, they're also working on all these other things at the same time. And that means that if you find the right question and the right problem in your model to work on, some of those problems and questions are very powerful because they tell you a lot about these other things. Also, if something goes wrong, it may mean, yeah, I've made some wrong assumption about what's happening in this specific system. But it could also be that we have an assumption at a much higher level that's wrong somehow. And just seeing that these things are connected is really important scientifically, because it tells you what the results mean. So if you learn something about this, you've learned something about a specific transcription factor. Either it's an exception or it follows a general rule. You may have also learned something about the general rule. You've also learned something about process of gene activation, either something specific or general rule. So this view of things has functions for both communication and research, because it also tells me how to communicate. If I'm a student, my students have to do an exercise which is to explain their work to their grandmother or their grandfather, and who are not scientists usually. And if I were to say, yes, grandmother, I'm working on the physical and chemical basis by which a transcription factor interacts with different molecules in order to activate genes in the conditions of metastatic cancer, that's going to be complicated. But you don't have to do that. You can start up here and you can say, well, you know, grandma, the reason why you're healthy or sick is because you have this little stuff in your body. You have cells and molecules and tissues and organs, and the smallest level is kind of the cells and molecules. And you know that we all come from a single egg, fertilized egg, and that egg produces all these different types of cells. And the way it does it is by using this, each cell has the same information, but different cells use different parts of that information, and they choose which parts to use because of these things called transcription factors. And I'm trying to figure out how one of those works. So you instantly put, again, I'm going really fast here because I want to finish our first part on time, but I'm trying to show you that this has a communication function. It gives you a route into a very complex story from a very basic level. And it also has a scientific function because it shows you how really specific details are related to large ideas. It helps you to find the right level to communicate to different audience. So with my grandmother, I need to start up here. With specialists, I can start right down here and say, you all know what transcription factors are. I'm working on NF-kappa B, and I'm not sure whether you know about its functions in this disease, but so it gives you an entry level for all different kinds of audiences. And it's also important for research because it shows that these layers of organization and models and structure are connected to each other. And it tells you what results mean, and it also can tell you where to look if something goes wrong and you don't understand. And that's where unexpected things happen. That's where good science happens. So I am finished with part one. I've given you my whole theory, and in the next part, after we take a short break, we will do some playing around with these ideas.
Sure. Thank you, Ras. I know for some people it could have been maybe too much theory, but we really enjoyed it, considering the fact that we talked about how much this communication is important for scientists and for science, really. So thank you for going thoroughly over this theoretical part. And yes, as you said, we will have 5 minutes coffee break now.
Okay.
And we will come back and do the practical part. We will do some breakout rooms and some exercise and a little bit more active joining from our side this time. So I know how difficult it is, and I'm still really sorry we couldn't do it in person. I know you, Russ, you're great in person. Yes, so let's do this coffee break, and we'll be back in 5 minutes, please.
Okay.
Okay.
Yep. I'm going to stop sharing, stop recording, and all kinds of things can go wrong. Hopefully not. And I'll see you back in a few minutes.